Have you ever wondered how scientists can read an entire human genome in just a couple of days? Something that once took decades and billions of dollars. Welcome back to the channel Bioscope. Today we are diving into the world of next generation sequencing or NGS, a technology that has completely transformed biology, medicine, and research. From unlocking the secrets of cancer to mapping the diversity of microbes in our environment. NGS is the powerhouse behind modern genomics. In this video, I will walk you through how NGS works, the different platforms, its workflow, applications, and what the future holds for this exciting technology. So let's get started and decode the future together. So, first question is, what is NGS? It is a high throughput sequencing technology that refers to advanced DNA sequencing that can process millions of DNA fragments in parallel. Compared to traditional Sanger sequencing, NGS drastically reduces sequencing cost and time, transforming genomics research and applications. If we talk about the Human Genome Project, it took 20 years and cost almost 3 billion rupees. But today, to complete the Human Genome Project, we only need 48 hours and we can do it just for $1,000. And the costs are still dropping. Applications can be spanned from personalized medicine to diagnostics, agriculture to forensics, evolutionary biology, and many more. Now, let's talk something about on which principle this NGS process is based on. The first one is fragmentation of DNA or conversion of RNA to complementary DNA or cDNA into small pieces. Adapter sequences are attached to fragment ends to enable amplification and sequencing. Then clonal amplification of fragments on solid surface or beads to create detectable signals. Then sequencing by detecting base incorporation events, for example, fluorescent, pH change, light emission, or probe ligation. Massively parallel sequencing, millions of sequencing reactions occur at the same time. There are some key features in this process, that is the massive parallelism, simultaneously sequencing of millions of fragments, then the high throughput, that is whole genome in a single run, base by base detection for high accuracy, and scalability from targeted gene panels to entire genomes or transcriptomes. There are major platforms and principles included in this process. The first and mostly used platform is Illumina. Here, DNA fragments bound to flow cell oligos, bridge amplification creates cluster, then incorporation of reversible fluorescently labeled nucleotide is imaged after each cycle. Then fluorescent tag is cleaved before the next base addition. For this platform, the advantage is it has very high accuracy, cost efficient for large scale project. And limitation is it can only sequence near about 150 to 300 base pairs. The next platform is ion torrent semiconductor sequencing. Here, DNA bound to the beads and result in the amplification by emulsion PCR. Nucleotide incorporation releases hydrogen ions that can alter the pH, which can be further detected by the semiconductor sensors. Advantage of this platform is fast, optics free, and cheaper. Limitation is errors in homopolymer runs. For example, if it has multiple adenine nucleotide, then it can make some error. Next one is Roche 454 pyrosequencing. Nucleotide incorporation releases the pyrophosphate generating light via luciferase. Advantage is it can read long base pairs near about 700 base pairs, and limitation is again the homopolymer error and the reagent cost. Next one is solid sequencing or sequencing by ligation. It uses fluorescently labeled oligonucleotide probe that ligate to the target DNA. Reads are interpreted by color space. Advantage is high accuracy and limitation is the complex data interpretation. The next one is the third generation or long read platforms. The first one is the PacBio SMRT or the Oxford Nanopore, which help in long reads and that resolve structural variants, repetitive regions. Direct RNA sequencing is possible here. Epigenetic detection without chemical conversion is also possible. But it has higher per base error rates, but it is improving right now. Let's talk something about the NGS workflow. In first, the sample preparation is done by DNA or RNA extraction. Then, if we take RNA, then we have to make the cDNA or complementary DNA, and DNA fragmentation is done. Secondly is the library preparation, where adapter ligation is done, optional barcoding for multiplexing, and PCR free options available to reduce biasness. In third step, bridge amplification or emulsion PCR is done. In the sequencing steps, platform specific chemistry is required that helps in synthesis ligation. After that, in the final stage, data analysis is done. First, we have to do the base calling, then quality control, then alignment, variant calling, and then annotation. Here you can see the workflow in the Illumina platform. You can also read the details on the Illumina website, where you can get more details about this library preparation, cluster application, sequencing, and alignment and data analysis processes. Next, we will talk about how we can control the quality for these particular steps. For example, magnetic bit purification that can remove unwanted fragments, and bioanalyzer or tape station that can help to check the fragment size distribution and concentration. The NGS process has many applications. For example, in whole genome sequencing, we can sequence the complete DNA blueprint. In whole exome sequencing, we can only do coding regions only. For transcriptome profiling, NGS helps in RNA sequencing. In metagenomics, that means microbial community studies can be done by NGS process. The chip sequencing helps in protein DNA interaction mapping. Targeted panels help to diagnose the disease specific things. In cancer genomics, we can use this technique to detect the mutation, and we can also profile the tumors. Non-invasive prenatal testing can be done, where fetal DNA from maternal blood can be taken out to study. Liquid biopsies that help to detect tumors or donor DNA in the blood. The NGS process has many advantages. That is, it is highly throughput and scalable. It is cost effective per basis. It detects rare variants, and it has many flexible applications. With this advantage, some limitations are also there. That is, short read platforms struggle with repetitive or complex region. It requires advanced bioinformatics infrastructure, high setup cost for instruments. Library preparation can introduce biasness. There are some future directions possible for this technique, that is cost reduction. For example, we can do it in $100 in genomes. integration with AI or ML, that means artificial intelligence or machine learning for faster interpretation, point of care sequencing for real time diagnostic. It can be personalized genomics integrated into healthcare, then expanded epigenetic and transcriptomic profiling that lead directly from native molecules. So that's all for this video. I hope you like it. Thank you for watching.